erythrocyte production and destruction. So some terminology for you. We have erythrocytes, which are mature red blood cells. They do not have a nucleus. RBCs is just an abbreviation for red blood cells. Erythroblast is a nucleated red blood cell precursor. It's an immature red cell that still has its nucleus. A normalblast is a developing nucleated red blood cell precursor, also known as an NRBC. We have a ruboblast or a pronormoblast. These are the earliest identifiable erythroid precursor cells in the bone marrow. That represents the first blast stage. So erythroid precursors, pronormoblast, are as mentioned earlier, they are the earliest erythroid precursor, also known as ruboblast. These are large cells with a high nuclear to cytoplasmic ratio, and they are deeply basophilic cytoplasm, so they have that deep dark blue cytoplasm. Basophilic normoblasts, this is the next stage of erythroid maturation. They are smaller than pronormoblasts with the intensely basophilic cytoplasm due to that abundant RNA that's still there. Polychromatic normoblasts, this is the intermediate stage with cytoplasm showing some mixed colors, so some basophilic and eosinophilic colors are starting to show through as that hemoglobin starts to accumulate. And this will indicate that hemoglobin synthesis is starting to begin. Criteria used in red blood cell precursor identification. So we will use the cell size, the nucleus, and the cytoplasm. So for the cell size, this will vary with the stage of development. Generally, it's going to decrease, the sizes anyway, is going to decrease as the maturation progresses. The nucleus, so you'll have a chromatin pattern. So immature precursors have fine open chromatin, and it starts to become dense and clumped once they start to become more mature. The size, so the nucleus size is going to decrease relative to cytoplasm as the cell matures. Then we have nuclear cytoplasmic ratio. This is high in early precursors, and this reflects a large nucleus relative to the cytoplasm, and that will also start to decrease as the cell matures. Nucleoli, these are present in early erythroblasts, and they disappear in the later stages. And then cytoplasm, so we look at the color. So we have basophilic, which is that blue. That happens early due to that high RNA content. And it becomes polychromatic with hemoglobin synthesis. And then finally that eosinophilic pink color in mature red blood cells. So a pronormoblast or a ruboblast. So we're going to go into a little bit more detail on each of those. So the nucleus in this takes up much of the cell, as you can see in that picture, that purple circle in the middle, that is the nucleus. So the nucleus cytoplasmic ratio is an 8:1. The nucleus is round to oval and it contains about 1 or 2 nucleoli. The purple-red chromatin is open and contains few, if any, fine clumps. Cytoplasm. The cytoplasm is dark blue because of the concentration of ribosomes and RNA. The Golgi complex may be visible next to the nucleus as a pale, unstained area. Pronormoblast may show small tufts of irregular cytoplasm along the periphery of the membrane. For division, the pronormoblast undergoes mitosis and gives rise to 2 daughter pronormoblasts. More than one division is possible before maturation into the basophilic normoblast. The location for the pronormoblast is present only in the bone marrow in healthy states. For cellular activity, the pronormoblast begins to accumulate the components necessary for hemoglobin production. The proteins and enzymes necessary for iron uptake and protoporphyrin synthesis are produced. This is also where globin production begins. The length of time in this stage, it's not very long. The stage lasts slightly more than 24 hours. All right, basophilic normoblast or prorubocyte. The nucleus, the chromatin begins to condense, starts to get a little more clumpy. It reveals clumps along the periphery of the nuclear membrane and a few in the interior. As the chromatin condenses, the parachromatin areas become larger and sharper, and the nuclear to cytoplasmic ratio decreases to about 6:1. The chromatin stains deep purpley red color, and nucleoli may be present early in this stage, but once they get later in the stage, they'll start to disappear. Cytoplasm, so when stained, the cytoplasm may be a deeper, richer blue than in the pronormoblast, hence the name basophilic for this stage. In the division, the basophilic normoblast undergoes mitosis, giving rise to 2 daughter cells. More than one division is possible before the daughter cells mature into polychromatic normoblast. Location, these basophilic normoblasts are present only in the bone marrow in healthy states. For cellular activity, it's detectable hemoglobin synthesis occurs, but the many cytoplasmic organelles, including ribosomes and a substantial amount of messenger RNA, completely mask the minute amount of hemoglobin pigmentation. Again, this lasts for only about a little more than 24 hours. Polychromatic normoblast. The nucleus, the chromatin pattern varies during the stage of development, showing some openness early in the stage, but becoming condensed by the end. The condensation of the chromatin reduces the diameter of the nucleus considerably, so the nucleus to cytoplasmic ratio decreases from about 4:1 to about 1:1 by the end of the stage. Notably, you need to remember there is no nucleoli in this stage. The cytoplasm, this is the first stage in which the pink color associated with the stained hemoglobin can be seen. The stained color reflects the accumulation of hemoglobin pigmentation over time and concurrent decreases amount of RNA. The color produced is a mixture of pink and blue, and that kind of results in a murky kind of gray-blue color. The stage names refers to this combination of multiple colors because polychromatophilic means many color loving. Division, this is the last stage in which the cell is capable of undergoing mitosis, although likely only early in this stage. The polychromatic normoblast goes through mitosis, producing daughter cells that mature and develop into orthochromic normoblasts. Location, so the polychromatic normoblast is present only in the bone marrow, once again in healthy states. For cellular activity, hemoglobin synthesis increases and the accumulation begins to be visible as a pinkish color in the cytoplasm. Cellular RNA and organelles are still present, particularly ribosomes, which contribute a blue color to the cytoplasm. The progressive condensation of the nucleus and disappearance of nucleoli are evidence of progressive decline in transcription of DNA. Length of time is a little bit longer, it's about 30 hours. All right, so orthochromic normoblast or metaerythrocyte, the nucleus is completely condensed or pycnotic or nearly so. As a result, the nuclear to cytoplasmic ratio is low. It's approximately 1 to 2. For cytoplasm, the increase in the salmon pink color of the cytoplasm reflects nearly complete hemoglobin production. The residual ribosomes and RNA react with the basic component of the stain and contribute a slightly bluish hue to the cell, but that will fade towards the end of this stage as the RNA and organelles are degraded. For the division, the orthochromic normoblast is not capable of division because of the condensation of the chromatin. The orthochromic normoblast location, it's present only in the bone marrow in healthy states. Cellular activity, so hemoglobin production is ramping up. It continues on the remaining ribosomes using messenger RNA that's produced earlier. Late in this stage, the nucleus is actually going to be ejected from the cell. The nucleus moves to the cell membrane and into a pseudopod-like projection. As part of the maturation process, loss of vimentin, which is a protein that's responsible for holding those organelles in proper location in the cytoplasm, it's probably important in the movement of the nucleus to the cell periphery. Ultimately, the nucleus-containing projection separates from the cell by having the membrane seal and pinch off the projection with the nucleus enveloped by cell membrane. Non-muscle myosin of the membrane is important in this pinching process. The enveloped extruded nucleus, called a pyronocyte, is then engulfed by bone marrow macrophages. The macrophages recognize phosphatidylserine on the pyronocyte surface as an "eat me" flag. Other organelles are extruded and ingested in similar fashion. Often, more small fragments of nucleus are left behind if the projection is pinched off before the entire nucleus is enveloped. These fragments are called Howell-Jolly bodies when seen in peripheral red blood cells and are typically removed from the cells by the splenic macrophages pitting process once the cells enter the circulation. This stage lasts about 48 hours. All right. Reticulocyte, nucleus. Beginning at the polychromatic erythrocyte stage, there is no nucleus. So your reticulocytes are the first stage that does not have a nucleus. The reticulocyte is a good example of the prior statement that a cell may not have all the classic features described, but may be staged by the majority of features. So in particular, when a cell loses its nucleus, regardless of cytoplasmic appearance, it is a polychromatic erythrocyte or a reticulocyte. For the cytoplasm, it can be compared with that of a late orthochromic normoblast in that the predominant color is that of hemoglobin, yet it still kind of has that bluish tinge to it due to some residual ribosomes and RNA. By the end of the polychromatic erythrocyte stage, the cell is the same color as a mature red blood cell, which is that kind of salmon pink color. It remains larger than a mature cell. However, so the shape of the cell is not the mature biconcave disc, but it is irregular in electron micrographs. Division, it lacks a nucleus, so it cannot divide. Location, so the reticulocyte resides in the bone marrow for about 1 to 2 days, and then from there it's going to move to the peripheral blood for about a day, and then it will reach maturity. So during the first several days after exiting the marrow, the polychromatic erythrocyte is retained in the spleen for pitting of inclusions and membrane polishing by the splenic macrophages, which can result in the biconcave discoid mature red blood cell. Cellular activity, so the polychromatic erythrocyte completes production of hemoglobin from a small amount of residual messenger RNA using the remaining ribosomes. The cytoplasmic protein production machinery is simultaneously being dismantled. Endoribonuclease, in particular, digests the ribosomes. The acidic components that attract the basophilic stain decline during this stage to the point that the polychromatophilia is only slightly evident in the polychromatic erythrocytes on a peripheral blood film stained with right stain. A small amount of residual ribosomal RNA is present, however, and can be visualized with a vital stain such as new methylene blue, and so called because the cells are stained while alive in suspension, or vital, before the blood film is made. The residual ribosomes appear as a mesh of small blue strands, a reticulum, or when more fully digested, merely just some blue dots. So when it's so stained, the polychromatic erythrocyte is called a reticulocyte. However, the name reticulocyte is often used to refer to the stage immediately preceding the mature erythrocytes, even when stained with right stain and without demonstrating the reticulum. All right, erythrocyte, fully grown red blood cell. No nucleus. It does not have a nucleus at this stage. The cytoplasm, so the mature red blood cell, is a biconcave disc that measures 7 to 8 microns in diameter with a thickness of about 1.5 to 2.5 micrometers. On a right stained blood film, it appears as a salmon pink stained cell with a central pallor area that will actually correspond to the concavity. The central pallor is about 1/3 the diameter cell. The erythrocyte, or normal, the mature red blood cell, cannot divide. It does not have a nucleus. The location and length of time in this stage, so mature red blood cells remain active in circulation for about 120 days. Aging leads to their removal by the spleen as described subsequently. So cellular activity, the mature erythrocyte delivers oxygen to the tissues, releases it, and returns to the lungs to be reoxygenated. The dynamics of this process are going to be discussed later on, but the interior of the red blood cell contains mostly hemoglobin, which is the oxygen-carrying component. It has a surface area to volume ratio and shape that it can enable optimal gas exchange to occur. If the cell was spherical, it would have hemoglobin at the center of the cell that would be relatively distant from the membrane. It would not be readily oxygenated and deoxygenated. So with the biconcave shape, even hemoglobin molecules that are toward the center of the cell are not distant from the membrane and are able to exchange oxygen. The cell's main function of oxygen delivery throughout the body requires a membrane that is flexible and deformable. So that is, it's able to flex, but return back to its original shape. So the interaction of various membrane components will be described later on. Red blood cells must squeeze through small spaces, such as the basement membrane of the bone marrow, venous sinus. And similarly, when a cell enters the red pulp of the spleen, it must squeeze between epithelial cells to move into the venous outflow. Deformability is crucial for red blood cells to enter and subsequently remain in circulation. Erythrokinetics. So, what is an erythron? I guess we should start with that as a definition. So the total population of red blood cells and their precursors in the bone marrow. Red cell mass is the volume of red blood cells circulating in the bloodstream. Then we have hypoxia as a stimulant for erythropoiesis, low oxygen levels or hypoxia will trigger the production of erythropoietin, which will stimulate and increase red blood cell production. Apoptosis, we've talked about before, but this is a programmed cell death that regulates the number of erythroid precursor cells to maintain balance and prevent overproduction. All right, so erythropoietin. Erythropoietin can be measured by chemiluminescence, although the reference interval for each laboratory will vary. 10 to 30 microliters is sufficient to maintain steady state erythropoiesis in a healthy adult. Increased amounts of erythropoietin in the urine are expected in most patients that have anemia, with the exception of patients with anemia caused by renal disease. So therapeutic uses, so we can use erythropoietin as a recombinant, and it can be used as therapy in certain anemias, such as those that are associated with chronic kidney disease and chemotherapy. It is also used to stimulate red blood cell production before autologous blood donation and after bone marrow transplantation. Unfortunately, some athletes have illicitly used erythropoietin injections to increase their oxygen carrying capacity of their blood to enhance their endurance and stamina, especially in long distance running and cycling. So the use of erythropoietin is one of the methods of blood doping. Aside from being banned in organized sports events, it increases the red blood cell count and blood viscosity to dangerously high levels and can actually lead to fatal arterial and venous thrombosis. All right, microenvironment of the bone marrow. So we have erythroid islands. These are specialized microenvironments within the bone marrow where erythropoiesis can occur. It consists of developing erythroid cells clustered around a central macrophage. So the role of the macrophage in erythropoiesis is it maintains homeostasis, helps to regulate the balance of erythroid cell production and destruction. Can promote tissue repair, so it can contribute to the repair of bone marrow tissue and support overall marrow health. It can secrete cytokines, so it can produce cytokines that create either a pro-inflammatory or anti-inflammatory environment. That depends on the need of what the marrow needs. Incretin systems, these serve as a physical scaffold for developing normoblasts, and it helps to facilitate their maturation and release. And then we have, red blood cell destruction, so hemolysis. So we have macrophage-mediated or extravascular hemolysis. This occurs outside of blood vessels, primarily in the spleen. So macrophages phagocytize and destroy damaged or aged red blood cells. This is typically a chronic process. And then we have mechanical hemolysis or fragmentation or intravascular hemolysis. This takes place within the blood vessels. Red blood cells are physically fragmented or lysed inside circulation, often acute, and can be a medical emergency. These can be associated with other complications like disseminated intravascular coagulation or DIC. It can also be associated with acute kidney injury or hypotension.